Shown here is a human adenovirus serotype 5's genome, and this is one of the most common actual vectors that is used in gene therapy, and here's the genome of it. And you can see it's really complex. It has a lot of coding regions, which is really why we debate whether viruses are living or not, because look how complex it is. It codes for so many things. Like, come on. But moving on from that, what I'm planning on doing is creating a gutless adenoviral vector, which means that it's missing everything, all of this except for the inverted terminal repeat regions, and a packaging signal. And it's going to have the rest of its space is going to be taken up by actual coding for other things. And in my case, I'm planning on putting Cas9 coding DNA and its guide RNA into tumors to help combat the KRas expression, which is an oncogene. But I'll talk about more about this plasmid first. So here you can see the Cas9 is from Streptococcus pyogenes, which is one of the most common Cas9s that's being used, and the guide RNA, which will target KRas. And KRas is a proto-oncogene, but mutations in it can cause it to become an oncogene, which means it's a cancer-causing gene. And by using the benchling software and having its CRISPR sort of guide go to work, I can see sequences that would be really good. For example, we can see the on-target and off-target score, and this is a sequence that I decided settling on because it has a pretty good on-target score and a really low off-target score, which is what we're really looking for. And that's also due to the fact that putting it through a blast, which basically means that I measured this sequence with every other sequence in the human genome to see if there are any duplicates. Because if this sequence and a PAM happens to occur in another part of the body, the CRISPR will also target that, which is not what we want. So that's what we're looking for. And also, for example, with this sequence, I also found another sequence that also has a PAM, and it's on chromosome 6. So that's definitely not something to go with. So for the guide RNA, I chose this to be the sequence that it binds to, and it checks. And the reason I chose KRas is because KRas is really, first of all, the RAS family itself is very responsible for cancer. It's present in almost 20% of cancers, and in other cancers, like in colon cancers or other cancers, it can be high as up to 80% of different cancers. And so the reason I'm targeting KRAS is because it encourages cell growth, and by shutting it down, basically you stop this sort of cell proliferation and this sort of undividing, like not undividing, but innumerable growth, which is the reason why cancer exists in the first place, because it doesn't want it to stop growing. And that's what KRAS is basically encouraging. And the reason I'm targeting, like I said, is to stop its expression. And there are a lot of things that we'll have to understand using this vector through a lab. And first and foremost, using a gutless adenoviral vector. It could also have other modifications to it, for example, changing the fiber knobs so it can bind to other proteins, so that can better bind to cancers. But what I'm thinking of is, like I said, targeting tumors that have their blood cut off so that this adenovirus doesn't actually spread throughout the bloodstream and target perfectly healthy cells who have their oncogene not turned on yet. So targeting tumors that have their blood supply cut off, and basically that will mean that the tumors will stop growing as much, and this will be really good for targeting tumors in their early stages where you can barely see them, not to the point where they're so massive that it's really just by surgical means that you can take it out. So yeah, this is what I've been working on, and this is hopefully I'll try to use this in a lab sometime soon after this whole entire quarantine thing blows over. But yeah, this is really a new frontier when it comes to being able to use CRISPR and actually having an adenovirus to put it into. I hope to explore different ways, different CRISPRs actually put in, like base editing and prime editing. So look forward to that. But this is what I've been working on, and I hope that it can be used. There may be some unintended consequences of actually using this Cas9, but we'll have to figure that out in the lab because this is merely just viewing and we can't really see much from it. But that's about it.